Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-level biology topics. This is biodiversity and conservation. Topic 4C1, biodiversity and endemism. Biodiversity includes genetic diversity between individuals within a species, as well as different species and in different ecosystems. Within an ecosystem, all organisms are going to be interdependent and they can affect the physical conditions around them. If there is rich biodiversity, it allows large-scale ecosystems to function and to self-regulate. Biodiversity enables further improvements by crossbreeding and genetic engineering. And if biodiversity is reduced within an area, the natural balance could be affected somewhere else. So having greater biodiversity is very important. Let's go to how biodiversity can be assessed at the species level. It's going to be measured using species richness, meaning the number of the different species within an area, as well as species evenness, meaning the number of organisms per species within a specific area. So if there are more organisms within a specific species, then we can say there is more evenness. Biodiversity can also be measured by looking at endemic species in an area. For example, you can look at kangaroos, which are endemic to Australia, and so on. So if there are more endemic species within an area, we can say there is greater biodiversity. Moving on to species abundance. So here we have an example, and again, this is from your textbook. If we look at two plots of land, there is plot A as well as plot B. They've given us a table here, and in plot A, we have 95% grass, 2% daisies, 1% dandelions, 1% buttercups, and 1% ladybird straw. And in plot B, we have 30%, 20%, 15%, 15%, 20%. We can see both plots of land have the same species richness, meaning they have the same number of species. However, the evenness is going to be different because we can see this is 95%, but the others have a lower percentage, yet this is more evenly distributed out. So we can say plot B has greater biodiversity in comparison to plot A. Topic 4C2, measuring biodiversity. Biodiversity can be measured using the diversity index, which is what we see here. D stands for the diversity index. The capital N is going to be the total number of organisms of all species. For example, here, this is going to be 40 because you add up all these organisms. And then the small n is the total number of organisms of each individual species. For example, here we have 4, here we have 12, here we have 3, here we have 4, and that's 17. And then, of course, this means a summation sign, meaning add up everything. So in order to generate data that can be substituted into this equation to find the diversity index, I need to generate the specific components. Since we already know this is going to be the total of all the numbers of organisms, it's going to be 40. So capital N minus 1 is going to be 39. And therefore, capital N into capital N minus 1 is 40 times 39, giving us that value 1560. Then to generate data for N minus 1, it's going to be 4 minus 1, giving us 3. 12 minus 1 giving us 11, and so on until 17 minus 1 giving us 16. So again, I'm going to multiply that times that to give us 12, that times that to give us that times that to give us 6, and then multiply to get 12, and multiply to get 272. Now I'm going to add up 12 plus 132 plus 6 plus 12 plus 272 as the summation, which is going to be the denominator, and then the numerator is going to be this data I generated here. So in the end, we have 1560 divided by 434, which gives us a diversity index of 3.6. So basically, that is how you measure the diversity index, but you need to remember this formula in your exam. Biodiversity can be seen in ecosystems that are very stable, in areas where there is high productivity, and in areas where organisms can grow and reproduce. However, in areas where there are extreme conditions like deserts, and areas that flood and so on, there is going to be lower biodiversity. And if there is lower biodiversity, it means ecosystems that have unfulfilled niches are going to be susceptible to incoming organisms that can rapidly overpower those that are existing. And actually, these incoming organisms can have a greater effect on the survival of the organisms that are already existing there, and therefore can affect or have an effect on biodiversity. This includes if they're predators and so on. Moving on to gene and allele frequency, and again, this is related to biodiversity. Allele frequency is the relative frequency of a particular allele within a population. When mutations occur, some are going to be harmless. However, some mutations can have damaging effects to the offsprings. We know that mutations increase the number of different alleles within a population, and hence they're going to affect biodiversity. If a mutation leads to an advantageous allele, there is a chance that it's going to increase in frequency because it's going to be selected for in case if there is selection pressure, meaning it's an advantageous allele and therefore it's going to be preserved within the population. For mutations that lead to lethal or damaging alleles, natural selection is going to remove those alleles from the gene pool, except if those lethal alleles give some little benefit to the organisms, then those alleles could be maintained. For example, sickle cell anemia, this is maintained in the human population because it gives some protection against malaria. Going to measuring genetic diversity, you need to remember that if there is no variation, populations can be completely destroyed. And again, without variation, if there is change in the environmental conditions, it could affect all organisms because they're going to have closely related DNA. So if there is variation within the DNA of different organisms, it can show health and well-being of a population they should be of. When measuring genetic diversity, usually we will look at the population of the heterozygotes for a given feature. So DNA is going to be analyzed. Only one band will show for homozygotes, but two bands are going to show for the heterozygotes, and this can be used to calculate the heterozygosity index like we're going to see in the next page. So the heterozygosity index is going to be the number of heterozygotes divided by the total number of individuals within a population. Using this worked example from your textbook, here we have a figure that shows 23 different organisms of the same species. For a single gene, we can see some of them are homozygous and others are heterozygous. This is heterozygous, that is heterozygous, that is heterozygous, heterozygous, and heterozygous because, of course, for the heterozygous we see two bands and the homozygous are going to show one band because the alleles are the same. So the heterozygosity index is going to be the number of heterozygotes, which is going to be 5, divided by the total number, which is 23, which gives us 0.217. And again, remember this formula in the exam. They can ask you to calculate a heterozygosity index like we saw with the diversity index. Moving on to topic 4C3, adaptation to a niche. Beginning with successful adaptation, I'm going to look at what a niche is. A niche is the role of an organism in a specific ecosystem or a specific community. If a species is successfully adapted to its niche, it means the individuals within that species have characteristics that increase the chances of survival as well as reproduction, and these are going to be passed on to the next generation, which are going to be the offsprings. Adaptations may be of many different types. There is anatomical adaptation, there is physiological as well as behavior adaptations, like we're going to see. Beginning with the first adaptation for survival, I'm going to be looking at fungal carnivores. Fungi are usually saprophytes, meaning they release enzymes that break down dead material in the soil. However, some fungi are active carnivores, like the nematode worms we're going to look at here. For this, they have adaptations that help them to capture and feed on these worms. They produce sticky nets or adhesive pads to trap the worms, and then some live inside those worms. So basically, they can destroy the worms in order to get nitrogen that is contained within them. And again, this is an assumption that they feed on these nematode worms in order to obtain a source of nitrogen. Looking at the camel, a camel is seen as an ultimate survivor. Camels, of course, are very large, and they have different adaptations which can be anatomical, physiological, and behavior. Looking at the anatomical adaptations of these camels, they have large eyes, and these give them good vision in all directions, and also they have long lashes that protect them against the sandy environment, meaning the lashes can protect the eyes so the sand will not go inside the eyes. Their nostrils are long and they are slit-like, meaning they can close to protect the sand from going into their nose when it's windy. Their upper lip is split, hairy, and extensible. This is going to allow them to identify good food and avoid thorns in order to protect them. So basically, it's protecting them against some of these desert plants that have thorns. Camels do have a hump at the back which contains fat, and this acts as an insulation against the heat from the sun. The fat concentrated inside there allows for easy evaporation of sweat over the rest of the skin surface, meaning there is going to be little fat insulation in other areas of the skin. So it's going to cool easily. They have large feet with tough pads, and these enable them to spread their weight as the camel walk to prevent heat damage. They have tough pads on their knees, and these are to protect them from the hot sand, meaning the tough pads are not going to be that much sensitive to the heat. Moving on to physiological adaptations of camels, we're going to go to thermoregulation, meaning controlling the body temperature. Camels do have supple skin which is covered in fine hairs, and these are going to be erected in order to trap insulating hair during cold nights. And again, remember, they live in areas where it's going to be cold at night and very hot during the day, so they should have some adaptations to protect them when it gets cold. Camels can withstand wide variation of internal core body temperature, and camels can lose 30% of their body weight through water loss and make it up in 10 minutes when they're drinking water. They also have a hump where most of their fat is going to be found, and this is going to be a food reservoir. Of course, fat can be respired in order to produce ATP. When we go to water balance, camels can withstand 30% loss in water without being damaged. They can drink up to 180 liters of water in 24 hours, and their osmotic balance is not going to be affected, meaning their cells will not take in water excessively in order to burst. Their kidneys are adapted to produce very concentrated urine. Of course, camels do have longer loops of Henle in order to produce more concentrated urine so that more water is going to be reabsorbed. And camels can produce dilute milk or continue to produce dilute milk even when they are dehydrated, and this enables them to successfully breed in the desert environment. Moving on to behavior adaptations of camels, they can sit down early in the morning before the ground warms up, and their legs are going to be tucked underneath so they absorb little heat from the ground. They can orient themselves towards the sun, so as little of their body surface area as possible is exposed to the heat rays. And again, this is orientation, so less part of their body is going to be exposed to the heat rays. They may lie in groups so that they minimize the amount of surface which is going to be exposed to the sun. And again, this can be when they come in groups, some of the areas like this part and that part are going to be shielded from the sun, meaning it's going to decrease the amount of heat they're exposed to. Also, camels can eat large range of vegetation, and this provides them with a lot of food sources, even with harsh conditions of the desert where there is little food. Going on to topic 424, gene pool and genetic diversity. We have to look at some definitions like a population. This is a group of individuals of the same species that occupy a specific habitat and a particular niche within that habitat. A habitat, of course, is a place where organisms live, and this can include the physical as well as the biological elements of their surrounding. The biological elements are going to be the biotic factors, and the physical can include maybe the water, the air, and so on. The total sum of all alleles within a population is going to be called the gene pool. Within a specific population of organisms at a particular time, the gene pool can vary based on the different alleles that are present within that population. Moving on to allele frequency. Allele frequency is the frequency of alleles as the environment changes through natural selection and adaptation. When selection pressure arises, these individuals that have the advantageous allele are going to survive, and their frequency is going to be increased. So this could potentially cause evolution. The frequency of a specific allele, for example, is going to be represented by P. This is going to be for the dominant allele, and the recessive allele is going to be expressed as Q. So here we can say P plus Q is equal to 1, and this is going to be the total of the two different frequencies. Moving on to using the Hardy-Weinberg equilibrium, this is P squared plus 2PQ plus Q squared is equal to 1. This can help us to find the frequency of homozygous dominant. It can help us to find that for heterozygous. Because it's going to be easy for homozygous recessive to be seen, we can use that in order to find the heterozygous, in order to find the homozygous dominant. So here we use the Hardy-Weinberg equilibrium, which states that a population that is not evolving, the allele frequency of that population will remain stable from generation to generation if there is no other evolutionary influences or environmental influences that cause changes. However, if a population is evolving, of course, the allele frequency will change from generation to generation, and so the population will not be in equilibrium. In an area when speciation occurs or when new species form, the gene flow will be reduced because there is going to be no more breeding between the two original populations. So that can decrease the allele frequency in some way. Here we look at an example of using the Hardy-Weinberg equilibrium. And again, this is extracted from your textbook. In this question they say there is the frequency of albinos in a population is 1 in 20,000. So I'm going to use this, which is 1 over 20,000, to give us 0.00005. This is going to be the Q squared, which is the allele frequency for the homozygous recessive. And I can use that to find the Q, get a square root of that, and you get 0.007. Then after, I'm going to feed that into this formula, P plus Q is equal to 1, in order to find P. So P will be 1 minus 0.007, and that gives me 0.993. This is going to be the equivalent of P. And then I can square that to find P squared. P squared is going to be that square, which is 0.986. So I can be able to find the frequency of the heterozygous, which is going to be 2PQ, and this is going to equal to 2 into P times Q, which is going to be 0.986 times 0.007, giving me 0.014, and that is going to be the frequency for the heterozygous. So if you know the entire population, 98.6% of the population is going to be homozygous, dominant for that specific feature, and 1.4 is going to be heterozygous, while 0.005% are going to be the homozygous recessive for the trait of albinism. The Hardy-Weinberg equilibrium has specific conditions. Of course, remember we said for a population that is not changing. Because it's not changing, that means there are no mutations. There is going to be random mating, meaning any organism can mate with any organism. And also it assumes that the population is very large. It assumes that the population is isolated, so there is no immigration or immigration of organisms in and out of that specific area. And of course, there is no selection pressure. This tells us that this is a highly hypothetical population. Moving on to the first condition, which is mutations. Of course, we know mutations or some random mutations are going to cause changes in the DNA of specific organisms. And when these changes occur, the allele frequency of the population is also going to be affected. The mutations that occur in body cells or somatic cells, these are not going to be expressed in the offspring, so they will just remain with the organism. However, if mutations occur in the germline cells, meaning those that produce gametes, then this is going to affect the next generation because that mutation is going to be transferred to the next generation or to the offsprings. Although we know mutations do occur, it's rare that they affect the population very rapidly. The next one is non-random mating. Of course, non-random mating takes place based on the Hardy-Weinberg equilibrium. It assumes that mating is going to be random, but actually in real life, it's based on what organisms within a specific population prefer. We know that if mating occurred randomly, then the frequency of alleles in the population will stay the same because random mating is required for stability of the gene pool. However, non-random mating is going to occur all the time because some features of the phenotype determine which organism is going to mate with which one. Some organisms in nature, before they mate, there is mating rituals, and some organisms prefer certain organisms with specific features, and therefore the gene pool is not going to be stable because there is going to be non-random mating. The next one is isolation. If the Hardy-Weinberg equilibrium is to be maintained, the population must exist in isolation, and this is going to be very rare in the living world because migration of organisms in and out of a population is going to occur, and this is going to affect the gene flow. It's either going to decrease it or it's going to increase it. And when there is very low gene flow between two organisms, sometimes they're going to change and become a new species. This is when speciation can result. So the key thing here is isolation is going to be rarely attained in the living world. Next, we go to selection pressure. Now, the Hardy-Weinberg equilibrium applies to areas where there is no selection pressure, because if there is selection pressure, it means the advantageous allele is going to be selected for. Of course, we know many alleles are going to be neutral, their effect, but some alleles code for a phenotype which gives an advantage or disadvantage. If the environmental conditions change, then a selection pressure is going to arise, and some individuals which have different combinations of alleles are going to have an advantage, and this advantage is going to be selected for, meaning the organisms that have it will survive and pass it on to their offsprings. So the genetic makeup of the population is going to be changed over time because this advantageous allele is always going to be preferred. So natural selection is going to occur, and this can cause speciation, meaning changes within the specific population. Next, we go to topic 4C5, reproductive isolation and speciation. Looking at isolation and speciation, and again, we're going to see the two different types of speciation later on. We know that when organisms are isolated, there is going to be less gene flow between them, and this can lead to speciation. Because the two isolated populations are going to experience different conditions, they're going to have different selection pressures, and if there is variation between them, certain features are going to be selected for based on the conditions or based on the selection pressure that is within that area, and those features are going to be preferred, or those alleles are going to increase in frequency within that specific population, and over time, there is going to be speciation that occurs. Isolation mechanisms can be geographical, meaning there is going to be a physical barrier. This could be a river, a mountain, and so on. This separates the two original populations. Isolation could be ecological, meaning they inhabit the same region, but they are living in different parts of the same habitat. It could be seasonal isolation. There is no gene flow because the timing between the members of that population is not the same. Meaning, for example, if some flowers are earlier than the others, then there is no interbreeding between the two, so the gene flow is going to be decreased. This is about courtship rituals, the mating patterns, and so on. So if they change, it means other organisms are not going to recognize that specific courtship ritual as well as mating pattern, and therefore they will not understand the language, or they will not understand each other, and they cannot be able to mate with each other in order for gene flow to occur. There is also mechanical isolation, and this could mainly be due to a mutation that could occur and maybe change the genitalia of the animals so they no longer feed, and this can lead to reduced gene flow, and so speciation can arise. Reproductive isolation is going to be the most important factor in speciation. And again, I said already, speciation is going to be allopatric or sympatric. I'm now looking at allopatric speciation where there is a geographical barrier. So if this is the original population, a geographical barrier is going to arise. Then the two are going to be isolated, and because they're in different areas, they're going to be exposed to different selection pressures, and they're going to adapt to those areas based on the variations they have. In the end, they're going to be two different species. Looking at adaptive radiation, and again here we're looking at Darwin's finches. Adaptive radiation occurs when species evolve rapidly to form several different species, which all fill different ecological niches. These were originally the same species, but over time, they have evolved to feed on different organisms. The shapes of their beaks are adapted for that specific food or for the food they feed on. And again, this is because initially there were variations in beak size or beak length, and maybe due to scarcity of food and so on, the different organisms started feeding on different food types. Organisms with the same shape of the beak were feeding in the same area. There is a higher chance that they got to mate and therefore produce offsprings, and over generation, that advantageous allele of feeding on that specific food has been selected for to become a new species. Going on to sympatric speciation, this occurs between populations of a species living in the same area. Like we can see, it's within the same area. There is no geographical barrier, but there is reproductive isolation. It could be mechanical, like we saw already. The genitalia do not match. It could be behavioral, like mating rituals. It could be seasonal, and again, animals that reach heat at different periods of the year, or plants that flower at different times of the year. So here we see there is isolation, meaning the specific organisms are going to be isolated within. They're in the same original area, but are they going to be isolated because there is no gene flow between them and the other population, and over time, they become a different species. Moving on to topic 4C6, conservation, why and how. Due to the increase in the global population, of course, we can see from about the 1900, the population is increasing exponentially. And due to the increase in the human population, there is more food required. We need more fuel because more energy is required. We need more areas to build houses, so we need more shelter. We need more raw materials and so on. And to get this, we have resorted to the environment, like cutting down trees to create more space for farmland, to create more space for construction, for schools, drilling of fossil fuels and burning fossil fuels to generate more energy. And all these have a negative effect on the environment, and they have led to climate change, as well as depletion of certain biological resources. So all these are how the human race is threatening biodiversity. A lot of habitats for organisms have been destroyed. A lot of plants have also been destroyed. So certain plants as well as animals are becoming extinct because of the human threat due to the increase in population and the desire for raw materials. Conservation is carried out in order to protect the environment as well as the organisms. It can be carried out in situ or ex-situ. In situ, this is carried out in the natural habitat, while in ex-situ, it can be carried out for plants in botanical gardens, as well as using seed banks, and for animals in zoos as well as wildlife parks. When ex-situ conservation is carried out, it enables genetic material to be conserved, and this ensures that the breeding population can eventually be returned to their natural habitat when the situation has been resolved. Ex-situ conservation usually occurs in the country where the species have been threatened. Examples of conservation include reclaiming land for use, reusing sustainable agricultural systems, protecting threatened species, as well as global legislation on pollution and greenhouse gases. Using ex-situ in plants, here I'm going to be talking about the botanical gardens. We can see different plants are going to be grown in order to keep them from being extinct. So they are provided with the appropriate conditions for growth, and when they are in these botanical gardens, it takes away the threat that almost led them to becoming extinct. And again, still in ex-situ, we're looking at seed banks. These are used to preserve seeds in a state of suspended animation, meaning they're not going to be germinated. To prepare the seeds, we will remove the seeds from the fruits. We can clean these seeds. Sometimes they use a self-antimicrobial that can remove microorganisms that are pre-existing, and then we can X-ray to ensure there is a developed embryo. Then we can dry, and this drying decreases the water content in order to minimize the chances of germination during storage, and after that we can store between -20 to -40 degrees to ensure longer storage time. Storage of seeds is carried out because many plants produce large numbers of seeds, and again these seeds are small, so many can be collected and stored in a smaller space. However, seeds from some plants don't store well, so these have to constantly be grown in field banks like plantations. However, these consume so much space and they require a lot of manpower, which is going to be quite expensive. Ex-situ conservation in animals, and again these are carried out in zoos as well as wildlife parks. They carry out captive breeding, meaning the animals are going to be captured and they're going to be bred in the zoos or in the wild parks in attempts to prevent their extinction, and then after they can be reintroduced into their natural habitat in order for them to thrive in those areas. The problems with captive breeding and reintroduction, of course, there isn't enough space and resources to put all the endangered animals in zoos and parks. It's going to be expensive, and again, it's going to be harder to maintain the conditions perfect for each of the animals. Also, when reintroducing the animals, sometimes it's unsuccessful because the original threat may not be completely removed, so the reintroduced animals could again be threatened because the threat was not completely removed. Another problem is when animals are bred in captivity, they're going to have a hard time adjusting to the wild because in captivity they're given food, they're given medication, but in the wild there is no food. They have to hunt or they have to gather food, and there is no medication, so it could be hard for them to adjust, and some end up dead. Also in the zoos, the populations are going to be smaller. There's going to be a smaller gene pool. However, this can be overcome, and again you have to remember this for your exam. The smaller gene pool can be overcome by using stud books, and these keep records of genetic data for breeding individuals so that an organism doesn't breed with the same individual over and over, because that will be decreasing genetic variation or genetic diversity. Also, zoos can swap sperm with other zoos to improve genetic variation. And another problem is the reintroduction programs for these captive bred organisms are going to be expensive. Lastly, we look at sustainability. And again, this is to overcome the problems associated with the human influence on the environment. This can include using sustainable agriculture. This is carried out using methods that minimize damage to the environment. And again, avoiding monoculture. Monoculture is when one crop is grown for over and over, and this is going to lead to fewer other crops being grown because there is one crop all the farmers are interested in. There is also use of more organic fertilizers instead of using the inorganic fertilizers, because these can dissolve in water and they can end up in streams and rivers, causing problems for the organisms that live in there because they over-fertilize, leading to algal growth, which can lead to eutrophication. Also minimizing pesticides, because these can destroy organisms or these can have side effects on organisms within the soil, as well as being washed away into the rivers where they're going to bioaccumulate within the organisms like fish, and those are going to cause problems. There is use of biological pest control which can be carried out. Of course, this is going to improve yield, and it causes no harm to the environment. To maintain sustainability, Some decision makers have to be involved, like politicians. They have to be educated on the benefits of sustainable methods so that they can carry out targeted spending in order to improve the conditions of the environment. And again, to attain sustainability, we have to carry out more research in the production of food and other resources that cause less damage to the environment. There are some countries like Costa Rica where tourism has been encouraged as a source of income, and in doing so, the people are working harder to ensure the biodiversity is maintained. You could also educate people through environment support programs to reduce pollution, protect animals as well as plants, and again, afforestation can be carried out in order to increase the number of the habitats as well as species of plants, and so on, over a longer period of time. So this brings us to the end of this topic. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.